The purpose of this video is to demonstrate a procedure we've developed in our laboratory for benchmarking the performance of our wide-field fluorescence microscopes so that you can implement the same procedure in your laboratory for day-to-day performance benchmarking. We have written the software portion of the procedure as a micromanager script so that it is available to any microscopy user. Micromanager is freely available software that can be downloaded from www.micromanager.org. Micromanager is fully integrated with ImageJ, a widely used, freely available image processing package developed at the NIH. Having downloaded and installed Micromanager, begin by starting the program. Then open the script panel, which can be found under the Tools button, and open the Performance script. The Performance script can be found at the website shown on the screen, and it can be cut and pasted into the script panel. This script can be executed by clicking the Run button at the top of the script panel. Even if you use some other control software for your microscope besides Micromanager, you can use this script as a template for implementing your version of this analysis in your microscope control environment. Upon starting the script, you will be asked which data to display and store. If you are using this for the first time, we suggest displaying and storing all of the data available. For day-to-day instrument benchmarking, you will likely develop your own protocols for storing and accessing the benchmarked data. You will be asked for the micron squared to pixel calibration for your instrument. Note, this is the area calibration, not the linear calibration, which would be microns per pixel. If you know this scaling for your instrument, you can input it directly. Alternatively, you can use a spatial calibration target. Here we demonstrate the use of a spatial calibration target to obtain the microns squared per pixel scaling. You will be prompted to place your spatial calibration target on the stage. Here we show the use of bright field illumination to focus on the calibration target. Once in focus, press OK, then depress the left mouse button, hold down, and select a distance between 2 points on the target. Be sure to note the distance between these points so that you can input this distance into the next dialog. Next, you will be prompted to place a fluorescent reference material on the microscope stage. If you want to calculate the reference parameters from a fraction of the field of view, select an ROI of interest now. Here we demonstrate the use of a 1-inch diameter piece of SHOT 475 GG filter glass as a reference material. This glass fluoresces in green wavelengths when excited with blue light. It is commercially available, highly photostable, and homogeneous. It is optical quality glass, so it is nearly defect-free. Once it is placed on the stage, focus on the surface proximal to the objective as we show here. Once in focus, move to any location off the edge of the glass, then click OK to begin the next phase of the benchmarking algorithm. Pairs of images at different exposure times are acquired during this phase to determine the exposure time when the detector saturates, and to determine the minimum exposure time required to detect fluorescence from the glass. Both the saturation and the detection threshold are benchmarked relative to the glass reference material, so the parameters can be compared when you use this piece of glass on another day. Once the saturation is determined, the stage is moved away from the glass so that background intensity in the absence of glass can be measured. Background fluorescence can come from intrinsic fluorescence or from reflections from the optical components of the system. The background intensity can also come from room lights if the system is not light tight. Upon completion of these benchmarking intensity measurements, the saturation, the detection threshold, and the linear dynamic range are determined. These intensity measurements can also be used to calibrate intensities between different experimental runs. These data, as well as the fitting parameters used to determine these parameters, are reported in a table. These data can be saved by clicking the File tab and using Save As. We suggest saving these data routinely to validate that your system is performing as expected. With these data, you can use control charting on your system to verify that the performance metrics are with an acceptable range.